Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really awesome guest who has been involved in creating a better tomorrow for so many people out there via the work he's been doing the last several decades. Today, we have the honor of being joined by Dr. Craig Venter, founder, chairman, and chief executive officer of the J. Craig Venter Institute, a nonprofit research organization dedicated to human, microbial, synthetic, and environmental genomics research. Also focused on the exploration of social and ethical issues in genomics. Dr. Venter, is widely regarded as one of the leading scientists of the 20th and 21st century for his numerous contributions to genomic research. He actually started out in the area of biochemistry, pharmacology, physiology at UC San Diego in the 1970s. In the 1980s, moved to NIH. There perfected his strategies for rapid gene discovery, and then in 1992, founded TIGR, the Institute for Genomic Research, where first in 1995 decoded Haemophilus influenza genome. In the 1998, he founded Celera Genomics, which ultimately led to the culmination in 2001 of publishing the human genome in the journal Science, and then went on to sequence fruit fly, mouse, rat genomes, various other genomes. And he's not stopping, continuing to go on, getting into really exciting areas of so-called environmental metagenomics, which is a topic we'll be discussing a lot today, especially that his new book just came out, The Voyage of Sorcerer II, the expedition that unlocked the secrets of the ocean's microbiome, where he highlights his amazing 15-year journey collecting literally tens of millions of organisms all around the world to further understand this resource. We're honored to have him with us. A lot to talk about. Dr. Craig Venter, thank you so much for taking the time out of your schedule today.
Well, it's great to finally be with you. You and I have chatted over the years, and finally got a chance to do something with you.
Absolutely. It's exciting. And, Craig, I've been meaning to ask you for a while, actually, because, when you go into the scientific literature, you published, I think I counted something like 90 papers or so on catecholamine pharmacology before you published your first genomics paper. And I was just been wondering, I had to ask, based on all the time you spent out there as a kid surfing on the ocean, or, per your first book, swimming with poisonous sea snakes and barracudas and stuff like that in Vietnam, did you never have any interest in sort of just old school, traditional grind and find natural product pharmacology from the ocean's biosphere back in the day?
No, that it never occurred to me. People always research things that they're interested in. I've always been an adrenaline junkie. And when I started my work at UC San Diego, nobody knew how adrenaline worked on cells, and they didn't know if it worked on the inside or the outside. My plan was actually to go to medical school and practice third world medicine like I was doing in Vietnam, because that was so rewarding. But for the first time, when I went from a community college to UC San Diego, I was introduced to high-end science and high-end scientists for the first time. And it's sort of like advising people today, you can't pick a career if you don't even know what's out there. And it's a world I didn't even know existed. Or if I knew about it, I knew I'd never be a part of it, being almost a high school flunk out. So, I think in part my Vietnam background, my medical background, impressed some of these scientists. I was actually given a research lab as an undergraduate and published my first major paper as an undergraduate. And I just got so excited about doing basic science and making discoveries. It's just a world that opened up to me, and I switched from medical school to going into science. So it's the physiology and pharmacology was this program. It was a multidisciplinary program at the medical school.
Right.
They wanted to have some kind of graduate program. Today it's called basic medical sciences. You have to do the first 2 years of medical school along with the medical students, and then go off to do something different. But yeah, I was just totally focused on the adrenaline receptor. Uh, it's I spent years trying to isolate it, proposing to do molecular biology. And then the first discussion in the mid 80s of the Human Genome Project came on, and
Right.
I was just immediately blown away and fascinated with it. I thought, here I've just spent a decade trying to get one gene, one
Yep.
protein, and there's this proposal to get them all. I said, I'd be crazy to not want to do that. And so that's how I made the switch.
Yep.
When Bob Lefkowitz, he was my major competitor on the adrenaline receptor. He got the Nobel Prize for the work, and I said that I found I couldn't compete with him, so I had to sequence the human genome. So he tells this story how he's now partially responsible for getting the genome sequence. So
Excellent. So, thinking back, I was in San Diego. I think the year was 2007 now, and you and Arnold Schwarzenegger gave back-to-back keynote addresses at Bio that year. And I don't remember what he talked about, but I remember your presentation really well because you put up I think a dozen or so slides of a lot of the things that were coming out of sort of the first leg of the sorcerer journey. I remember the one really cool bug that was spitting out hydrogen or some other type of fuel. And it was really very captivating presentation. I'd love if we could go back to 2003 because you're doing a lot of stuff at that time, the dog genome, the rat, you're sequencing malaria. Talk a little bit about what got you out on the ocean, thinking about metagenomics at that time, into the Sargasso Sea, and a little bit of what leads up to Science 2004, environmental genome shotgun sequencing of the Sargasso Sea.
It's a great question because it was a lot of different pieces coming together.
Right.
So back in 95, when we sequenced the first genome, we had to write the algorithm for trying to assemble the genome. That was done by a team led by Granger Sutton at my institute. We didn't really know how good the algorithm was going to be, and we're doing this experiment real time. We didn't start out to do the first genome in history. We were just testing this idea of whole genome shotgun sequencing and testing whether the algorithm and all this stuff would work, and it worked fantastically. We went on to do other genomes early on. So after the first one was so successful, everybody in the world wanted to send us their genome to sequence. And the DOE gave us a huge grant to fund all these environmental genomes. We did the first archaea with Methanococcus as the third genome that we did. But we got sent a clinical isolate from Norway of a patient that was refractory to treatment, and they asked us to sequence the organism. And the nice thing about the algorithm, and one of the things I really like about genome science, in contrast to the field of biochemistry, when I was in biochemistry, people were successful by being able to tell a good story. And quite often, the stories outlive the data. And I didn't like that loosey-goosey aspect to it. So I was looking for something absolute. With DNA sequence, you either have it or you don't. It's either accurate or it's not. And so with these algorithms, they were like truth serum. For example, there was a huge argument as to whether cholera had one or two chromosomes. And different labs were doing experiments debating on it. We sequenced the cholera genome, and it clearly assembled into two separate chromosomes, and it settled the argument. And it's because the algorithm was that good. And the same with this Norwegian sample. Instead of one genome, it assembled two clinical isolates totally separately. We had no idea what was there, and it showed me how powerful the mathematics was. And that's where I developed the notion that each species, each one of us, has a unique mathematical solution for our genetic code. And so at that time, I knew we could definitely deal with complex samples. It wasn't clear how complex. So after I left Solera, I took a brief sailing sabbatical. I sailed down to the Caribbean and lived in Tortola for 3 or 4 months, recovering from all the effort for sequencing the human genome. I've always been a sailor, a swimmer, a surfer. And this paper came out. I was still reading the literature, even in the Caribbean. And this paper came out in PNAS talking about how little diversity there was in the microbial part of the ocean, and that all the diversity was on land. And I thought, this doesn't make any sense to me at all. And so I was just thinking, I thought of this experiment of taking a sample off of Bermuda, because that's where most of the organisms were being characterized. And microbiology at the time was you either could see the organism under the microscope or you could grow it in culture. If you couldn't do either one of those, it basically didn't exist. So they numbered the organisms that they were discovering in the Sargasso Sea one at a time, and they were up to SAR 11, Sargasso Sea 11. Yeah. And so, again, that was contributing to, probably people thinking there was low diversity. It just didn't make any sense. So we decided, I knew Tony Knapp, the head of the Bermuda Biological Station. Bermuda was a major stop-off sailing point for me to and from the Caribbean, and I love it there. So we decided just to take a barrel of seawater, filter out all the microorganisms on these tiny millipore filters, and just isolate all the DNA and sequence everything. And that led to our major paper that just blew our minds and everybody else's minds. We discovered thousands of new species that people had never seen before. We stopped sequencing after, I think it was 1.5 million new genes. Yeah. And it just totally changed the paradigm instantaneously. And you can't talk to a biological oceanographer that doesn't know that paper and that study today. Yeah. So the question was, in the way oceanography worked, people set up stations where they repeatedly sampled at these stations. And that didn't make any sense to me. As a sailor, you have currents, the water is always moving. Right. So a fixed geographical point on land makes sense. But in the ocean, it's like you're sampling in the middle of the river. It doesn't tell you anything other than how the river is changing. It doesn't tell you about that point in time. So instead of thinking about sampling over and over again in Bermuda, like oceanographers wanted us to do, I thought, well, what happens if we take a sample 200 miles from here? Is it going to be the same? If it's different? Maybe some of the organisms that were really rare in our sample off of Bermuda might be more abundant somewhere else. And so that was the start of the Sorcerer expedition. Surviving a year in Da Nang, Vietnam, dealing with the death of thousands of people my age and younger was a traumatic year to get through. One of the notions that kept me alive was that if I survived, I wanted to sail my own boat around the world.
There you go.
I was reading about sailing. So I thought, why in the hell not combine two things that I like? ocean sailing, and discovery science.
Yep.
And I did a little reading. So there in the 1870s was the Challenger expedition.
Sure.
It was the first actual scientific expedition. Darwin was an observer on a survey ship. Most ships carried a biologist of some type. But the Challenger was the first actual expedition. And what they set out to do, they started in Halifax. Every 200 miles, they dropped a dredge to the bottom of the ocean to see what they would find. So the notion was in the 1870s, the first transatlantic cables were being run. We knew nothing about what the bottom of the sea looked like. There was some crazy theory. And, I argue most science discoveries are undoing crazy theories from the past. This was the same situation. Biologists decided there was no way you could have life below, I think it was 16,000 feet in the ocean. How one comes up with such a notion, I have no idea, because oxygen runs out a whole lot earlier. But at every depth, every 200 miles, the Challenger found new species. They were looking at macro species.
Right.
But, they discovered the Mariana Trench, the deepest parts of the ocean. Everywhere they looked, they found life forms. And so that changed the notion of that. And I thought, this is fantastic. We could do the same thing on a modern sailboat, looking at the world that Darwin couldn't see, that the Challenger scientists couldn't see, because we had new tools. We had the tool, not of microscopes, but of DNA sequencing. So we could characterize things simply by sequencing their genetic code to find out who's there. So we decided to do the same thing, take samples every 200 miles. You may ask, why 200 miles? That's how far a decent-sized sailboat sails in 24 hours.
Okay.
So you basically stop once a day and take a sample. And we followed largely the route that the Challenger did, with some exceptions. But the most amazing thing was every 200 miles, 80% of the sequences were totally unique. I mean, it just shows, the ocean, instead of being a giant homogeneous soup, was millions and millions of microenvironments.
Yep.
And, maybe every sample, every mile, we would have found those kinds of differences. Because, if you go out in the ocean, there's temperature gradients, there's current gradients, there's sunlight gradients. Some water's cloudy, some is crystal clear. It's not surprising that there's microenvironments, but I think it was a little surprising to see the microbial diversity was that diverse. And so we continued that around doing a circumnavigation. Then we got back, we covered both the West Coast and the East Coast. We sailed up to Glacier Bay in Alaska and sampled from major glaciers. Then we sailed back across the Atlantic, covered the Mediterranean Sea, the Baltic Sea, and went up through the Bosphorus into the Black Sea. So we covered a lot of mileage, 65,000 miles. But it wasn't just taking scientific samples. We had lots of adventures and got arrested at least twice, and got caught up in the politics of science.
Sure. I know that well. Yeah, let's talk about some of the events beyond the sampling, because one of the one area that your institute has been publishing a lot on last couple of years has been the microbiome. I know you were a collaborating institute with the NIH Human Microbiome Program, and you published a lot on sort of what we're learning about the connection between the human microbiome and various diseases, cancer and fibrosis and inflammatory bowel disease, what happens when we screw that stuff up, dysbiosis and so forth. it was really interesting because when you went through the Panama Canal, you stop, I think somewhere near the Smithsonian Tropical Research Institute, and you do a little bioprospecting. You don't go tearing up the rainforest or anything, but you sample some of the bugs right at the intersection, sort of where the water and the soil connect.
Yeah.
And I was like, wow, that's like a major ocean microbiome connection there, just like the human one. Well, what are some of the things you hope to learn at that interface?
So, in fact, the human microbiome was born at the same time. We did the first experiment of not just shotgun sequencing off of Bermuda. We decided to do a shotgun sequencing of the first microbiome. So my institute did both environments at the same time.
Right.
But this incredible diversity. We made we converted some astronomers and physicists to join us because we found more microorganisms in the ocean than there are stars and planets in the universe.
There you go.
By several orders of magnitude. So they decided, they should be looking in instead of looking out. It was a far more diverse universe that we had. And that's just it. The diversity was so phenomenal. Trying to understand the interactions of the different types of metabolism. When there's sulfur in the soil, the microorganisms metabolize sulfur. With the Department of Energy and BP, we sampled deep inside the Earth, over a mile deep, in some of these coal bed methane mines.
Yep.
And one of the things we found in the oceans was not just single punctate organisms, but clouds of very similar organisms, as much as 50% sequence diversity, but largely the same order of genes.
Okay.
And so instead of Sargasso Sea 11 being a single organism, it's part of this cloud of maybe millions of related organisms. And so the two major drivers of evolution, in my view, in the oceans at least, are sunlight, so UV light, and oxygen. Those are the two major mutagens. So here, these photosynthetic organisms that provide half the oxygen we breathe are constantly bombarded with UV light. That's where they get their energy from light, from sunlight, and they're in a high oxygen environment all the time. So they're constantly mutating and forming new versions of themselves. We found just the opposite in the deep earth.
Okay.
In the deep earth, so there's no oxygen, and clearly there's no sunlight a mile down, and the temperatures are pretty warm, around 100 degrees centigrade. And so what we found was a lot of diversity with individual organisms, but one of the organisms, the closest relative we found, was in a volcano in Italy. So it could be the intense biodiversity beneath the earth, maybe interconnected across our planet. Because there's nothing driving evolutionary diversity in the same way. These are much more ancient organisms, but they live off of sulfur, or they convert CO2 into methane. There's so many different metabolic pathways, but it was striking how different it was from the ocean, and how different from our human microbiome. So it was like biologists envisioned the ocean was lots of individual organisms, so good diversity, but no variation around that diversity, and species being conserved perhaps across the planet. So, very, very different environments, very different worlds, but you can't go anywhere on this planet and not find life.
Right.
So the highest temperatures are over, I think, 120 degrees centigrade. So if you go deep enough into the earth, it's too hot for life. But we have organisms that live on boiling water temperatures. in high acid or a base. If you put your finger in it, it would dissolve.
Hmm.
These organisms live happily in those conditions. So the diversity of biology that we found, and the true is the rainforest, they were metabolizing things totally different than the what we found in the Panama Canal, just a
Yeah.
short ways away.
Right.
So it's the name of the game was diversity, and it's so much. We don't bother trying to name things. I mean, we're at hundreds of billions of organisms.
Right.
Um, they're best characterized by their genome sequence.
Yeah. And continuing along that diversity theme, in 2017, you published two fascinating papers back to back. So in February, you published this paper on the Baltic Sea virome, diversity and transcriptional activity of DNA and RNA viruses. And to sum it up, there's a lot of viruses there. The next month, you published this really cool paper on the DNA virome in human blood. Again, we're awash in unknown viruses. And it gets me thinking about these sort of good virus themes. Obviously, we hear all about the bad stuff, but you pose, a series of philosophical questions in the book, and one of them is, are the viruses in charge? Whether we're thinking about topics like bacteriophage therapy in sort of this area of antimicrobial resistance, or just all these cool viruses that, like hepatitis G, which we profiled on a show a little while ago, that makes us resistant to other viruses. Talk a little bit about, because I mean, the diversity here is amazing. I mean, there's the number per liter, whatever, the 10 to the 10th there. I mean, it's crazy. But talk a little bit about your view on these viruses and some of the potential applications here, because I think it's
So for every bacterial cell, there's 10 viral cells out in the environment. Then the ratio changes in different parts of the world. 99.9% of viruses are not harmful to humans.
yeah.
We hear about the ones that are for obvious reasons. And the viral world is totally unknown. I mean, when we sequence the viruses in the ocean, we sequence the ones in the human bloodstream. It's just chock full of viruses, but the sequences don't match anything else because so few viruses have been characterized, because the focus has been on disease-causing organisms. So that's why if you look at the imbalance in the literature, you think, well, viruses are clearly bad. They must all cause disease. But for everyone that causes diseases, there's thousands that regulate the copy number of the different microbes in the ocean. That's what controls life from getting out of hand. But the most amazing thing that we discovered about viruses is that not only do they not want to kill their host, viruses will bring in new copies of, for example, photosynthetic enzymes,
Yep.
basically upgrading the software in their host. So not only do they not want to kill their host, they want their host to thrive and do well. So that's why I was talking about the issue of who's really in charge of, viruses are keeping cells alive and they're keeping us alive. It's a strange biological interdependency kind of hierarchy with, viruses aren't just there to kill organisms. As I said, if they kill off their host, they go out of existence.
Right.
So it's an amazing relationship of how the DNA software gets constantly upgraded. And viruses in our own bodies exchange genes. We don't know if they're improving our software or not, but nobody's really looked at that before we made this discovery in the oceans.
Yep. Yep. Craig, there was well, it wasn't fun for you guys, but there was this episode. I think it's when you were leaving New Caledonia, I think maybe headed off to Australia, and you all eat some bad fish. Well, the fish wasn't bad. The fish ate some bad stuff,
Yeah.
namely this sigma toxin
Yeah.
produced in these dinoflagellates. You also talk about this other neurotoxin later on, this domoic acid. Obviously, these organisms have the ability to create really interesting substances to defend themselves. That's what, they defend themselves with chemistry. And it gets me, coming back to sort of pharmacology for a minute, it gets me thinking about sort of all of sort of our current pharmaceutical armamentaria that is based on venoms and poisons, obviously things like Botox, the ACE inhibitors, the original GLP-1 drugs. A little bit of thoughts as you assemble, as you call it, these design components for the future. Applications for, it's not always easy to have a bunch of black widows around or cone snails, but the use of potential sort of pharmaceutical discovery of some of these materials.
No, it's a great question because the chemistry we discovered looking at the genomes of all these organisms can do far more complex chemistry than our best chemists can. They make very complicated molecules. It's part of the defense and the biological warfare that's going on between organisms out there. That's where a lot of antibiotics come from, right?
Yep.
They secrete molecules to try and kill off some of their competitors. So there's groups that have tried to harvest things directly from the ocean. My notion is to take this huge library we have of genome sequences and pathways and put those in synthetic organisms like we made
Yep.
the first one of, and create whole new chemical libraries
Mm-hmm.
that are far more complex than our best chemists can make. But
Yep.
then these same cells could be the basis of manufacturing
Yep.
chemicals. So this limited repertoire we have as a pharmaceutical society right now could probably be expanded one or two orders of magnitude by using the biochemistry of the planet.
Yep. Yeah, I like that production side of it as well, because that's one of the things that's always hindered natural products, even though it's the basis for most of the industry, the ability to make these complex molecules at the same time. I think that's an awesome connection. Um, let's talk a little bit about the extremophiles, if we because I saw you on Brian Keating's show a couple of weeks ago. You were talking about Deinococcus and its ability to reassemble its genome. You published this really cool paper. This was back in 2010 on the Hydra and their genome, and they have this really cool ability to dedifferentiate and transdifferentiate itself back into a youth. And then, of course, I think about the tardigrades, which is part of the whole DARPA biostasis program now. That they're pretty good at protecting their proteome from all sorts of insults. Um, I mean, obviously, these all have connections to what we'll call resilience and robustness. I mean, these organisms hang in there through thick and thin. Talk a little about your thoughts in terms of applications and potential human longevity. I mean, obviously, I know you've added Human Longevity Inc. is more of a sort of a prevention story, but obviously, some of the knowledge that comes out of how these organisms hang in there is probably pretty important for us in other fronts as well.
It's, developing resilience and developing mechanisms for survival is the history of biology on this planet. That's why these microorganisms exist in every niche imaginable on the planet. As we talked about, from strong acids, strong base, high temperature, cold temperatures, high doses of radiation. So with Deinococcus radiodurans, we sequenced that sometime in the 90s, I think, and it's
Yep.
the most radiation resistant organism. In fact, I think of a Japanese group tried coating some on the outside of the space station and found that it survived for years out there without any difficulty at all. I got the head of NASA at the time annoyed with me because I knowing what they did in the space station, I said the outside of the space station is covered with human feces. So we're already doing this experiment.
There you go.
And it turns out they just, open a little valve and pump stuff outside the space station, which it didn't go very far. Sort of, stuck to the outside of the space station. So I wasn't wrong in my description. And what they do now is they package all the human waste into stainless steel containers and launch them back into the atmosphere to burn up. So I tell people to be very careful when they're wishing upon shooting stars,
Right.
that they could pick a bad one.
Yeah. So, coming back to the issue with biodiversity and sort of intellectual properties, I was couple careers ago, I was involved in this project with Pfizer. They were developing various ethnomedicinal plant-based drug products, and they ran into some trouble with this cactus from Namibia because they pissed off the sand bushmen and the conventional biologic diversity and all that stuff. And there was a bunch of issues, let's just say, in developing the drug. But thinking about as you mentioned, you were boarded, you were threatened with sinking and so forth at different parts of the trip. And it got me, as you were just mentioning before, there's a lot of currents out there. And if you're collecting microbes from wherever in the South Pacific, how do you know that the stuff didn't come from New Zealand or Chile or somewhere else? I mean, it's a pretty big ocean out there. Say a couple words about it, because it seems like, you're making these amazing discoveries that most likely are not going to be made if you're not taking those samples. But clearly, people got angry with you a couple times.
So it's not a problem. It's not a problem that Darwin had because there weren't international laws then. In fact, he's lucky PETA didn't exist. Some of the experiments he did on the iguanas, he wanted to see if they could really breathe underwater. So he'd tie rocks to them and throw them in the water and see if they could survive. If I did experiments like that, I'd probably be hounded out of science. But since then, there's this thing called the law of the sea, so that it's been extended now to genetic sovereignty. So countries own all the genetic organisms, certainly within 3 miles, but it seems to extend out in most places to 100 miles off their coast.
Yeah.
So, but out in the open ocean, more than 100 miles away from the coast, those organisms belong to nobody or to everybody. But as soon as they flow across the 100-mile boundary off the Marquesas Islands, they become French genetic heritage.
Yeah.
And there's roughly a 1-knot current. So when they flow into French waters, they're French genetic heritage. When they flow back out again, they lose their Frenchness. And so it's understandable how these rules developed, primarily defending fishing rights, so that these major ocean-going trawlers wouldn't come in off of, a mile off the coast and take all the fish from a local country, even though they still do that when they get away with it. But these laws were first to protect fishing, and then with pharmaceutical companies like you're describing going into the Amazon and other places, people thought, well, biologists are just here to discover billion-dollar molecules at our expense. You might even remember when we took the first samples in Bermuda, even though Tony Knapp had research permits as head of the Bermuda Biological Station. There's this big round of news stories how we were stealing Bermuda's genetic heritage and their future livelihood. Well, I don't think all the banks in Bermuda even know about microbes off their coast. But so this developed into a huge problem, and we had a whole team that had to work with governments from every country we wanted to visit or even pass through their waters 100 miles offshore to take a sample of seawater. It seems pretty silly, you know, if we just did it, nobody would notice. But the fact that it was me doing it, everybody noticed. And so we had to work with their militaries, get permits. And sometimes even those permits weren't enough. And we got caught up in geopolitics. So when we sailed from the Galapagos to French Polynesia, when we first entered the Marquesas, we were placed under house arrest by the French.
Right.
Even though we had permits, the Tahiti government was in a battle with Paris as to who controlled the waters in French Polynesia. Was it the local government or was it the French government?
Mm-hmm.
And so we got caught up in the middle of that. The French told us if we left harbor, they could not guarantee our safety, which was more than a veiled threat. Not too much earlier, they had sunk the Rainbow Warrior in Auckland, New Zealand harbor.
Yeah.
And what did the Rainbow Warrior do? It protested the French nuclear test in the South Pacific. So part of what we learned was that the French were certain that I was going to go to the atolls to take samples to prove how much genetic damage they caused. But fortunately, I had a satellite phone. I knew the French ambassador. You know, I'd been to lots of things at the embassy. But ultimately, we had to get the U.S. military involved and the State Department involved. The U.S. Navy had to contact the French Navy to tell them that we were a U.S. research vessel under U.S. Navy protection and to back off.
Nice.
And so that was comforting to have, you know, big brother on your side for once.
Yep.
But I still had to fly to Tahiti to negotiate another MOU with the Tahitian government for sampling there, and that we'd create a joint database. So the geopolitics really got frustrating. And as a result of all this, we took very few samples in French Polynesia, because by the time the permits finally got approved, we had to move on and get down to New Caledonia and in Australia. We also got arrested by the British, so it's not just the French. So we were sailing across the Indian Ocean, and there's this famous set of islands, the Chagos Islands.
Yep.
They're very famous in sailing literature, where sailors crossing the Indian Ocean stop there. And it's one of the most beautiful coral atolls in the world. And I notified the State Department directly that we were going to stop there just for a break from ocean sailing, but we were not going to take any samples. So Chagos Islands include Diego Garcia, where the U.S. has its major B-52 air base that it rents from the British. The problem was the British thought my notoriety and all the attention the expedition was getting would bring attention to the Chagos Islands, and the fact that the British had rounded up all the natives and kicked them off their own islands for British and American military bases. And the natives sue in world court every year. They win, and the British are supposed to let them have their land back. But the caveat is once they don't need it for military purposes. So they thought I was going to bring attention to all this. And so they're in their brilliance, they thought by arresting me, that would not bring any attention to anything. So we were actually boarded by a dozen heavily armed military in a rib They had no insignias on uniforms, no flag. They could have been pirates for all we
Sure.
knew, but we were outgunned. So they boarded us, took our passports, notified us that they were, part of the British military. I went down to my cabin and I happened to know the U.S. ambassador to the court of St. James, England, and the UK. And fortunately, he took my call right away. And I said, we're being held at gunpoint in the middle of the Indian Ocean on Chagos Island for no apparent reason. I said, we notified the State Department we're stopping here and that we're not taking any samples. They have no basis to hold us, but they're holding us at gunpoint and won't return our passports, and said that they had a right to impound the boat and arrest us. So he said, hang on, I'll get right back to you. So he obviously went to the head of the British government. I filed a formal complaint that they had no uniforms or insignia, that they just looked like a group of pirates. So the next day, when they left one guard on board, next day when they came back, they were in full formal military dress with the dress battle flag, and so apparently I was getting through at some level.
Yeah.
And they brought an agreement for me to sign, confessing to breaking this law from the 1800s of having unregistered dive equipment aboard a sailing vessel.
Mm.
And I said, really? That's all you can come up with? And they said if I didn't sign it, they would leave the agreement with me to think about, and they would come back once a week to see if I was ready to sign. Obviously holding us hostage there.
There you go.
So the ambassador was on the satellite phone with me. And so I said, Mr. Ambassador, I'm going to sign this, but I'm signing it under duress. So the military captain had no idea what that meant. I signed the agreement. They wouldn't give me a copy of it, but they gave us our passports back and said we were free to go. So we pulled up anchor. We got about a mile outside the hotel, outside the atoll, and my satellite phone rang, and it was the ambassador. He said, "Stop immediately, or they'll sink you."
God.
And I said, "For God's sake, why?" And he said, "The British government will not accept an agreement signed under duress." So he goes, "I want you to think very carefully before you answer this question. Were you under duress when you signed that agreement?" And the thought going through my brain was, well, I'm really fucking under duress now, because if I say yes, they'll sink us or arrest us. So I said, Mr. Ambassador, I was not under duress when I signed it. He said, that's the answer I was hoping for. You're now free to go.
There you go.
So that was our episode for having a friendly stop in the middle of the Indian Ocean. Nothing to do with science, nothing to do with sampling, just getting caught up in geopolitics.
There you go. There you go.
So they don't make science easy in the world anymore.
No, definitely not. Oh, let's talk about a couple other topics now. One which, I guess maybe a little contentious. I guess we'll call it marine geoengineering. And I'm thinking it's about a year ago I had Frank Marks on the show. He used to head up hurricane research at NOAA, and he was talking about this program they had. It used to be called Storm Fury, and it existed between like the early 60s and the early 80s, basically figuring out how potentially you can modify a hurricane by dumping all sorts of things like silver iodide and various cryogenic materials and hygroscopic materials into the water and the clouds. we hear things about things like iron fertilization of the oceans to increase productivity of these organisms, to suck more carbon dioxide out of the air and so forth. Your thoughts just on the general area of marine geoengineering, whether it's with foreign substances or with some of these potential synthetic bugs that you can whip up that can do these things maybe 10 times better than the current bugs.
I'm very clear on this. I think geoengineering is one of the stupidest things that we could do to our planet. We're messing it up pretty badly as we're going. But you can see with these hundreds of millions of organisms, it's a very complex ecosystem that's very dynamic. And so they thought, well, in some parts of the ocean, there's less iron. These organisms need iron. So if we just seed the oceans with iron, we'll get a lot more photosynthetic organisms. They'll capture a lot more CO2 and everything will be great. But nobody ever thinks of the consequences of these events. So geoengineering, and there's so many different aspects of this that, like everything, they sound really intriguing. Why don't we introduce a new predator into an environment to get rid of the smaller predator that's eating our crops or something? And our ecosystems are in a very delicate balance.
Right.
And it's very easy, as we've seen over and over again throughout history, of making these introductions and having all kinds of unforeseen consequences. We sailed into multiple mile wide seas of plastic and garbage. We're doing geoengineering by putting all our trash in the oceans.
Right.
And waterways, and that plastic breaks down. So now there's microplastics everywhere, even supposedly in bottled water and things that are pure, these tiny fragments. We've been isolating some of the organisms that break down plastics, but the problem is some of them break down to toxic chemicals, not to just they disappear and get metabolized.
Mm-hmm.
So we do much better to un-geoengineer by cleaning up all the trash and the plastic. And there's a lot of places trying to do this.
Sure.
The amounts are just so vast. It's unbelievable. I had a trip several years ago with the head of of health in Mexico, visiting Chagas and the Chagas River. And they showed how just, tons and tons of plastic bottles and stuff come down the river from the neighboring countries, and the river just gets totally clogged with trash. This is happening in all our different environments. So, until you could do enough safety testing, which I don't know how you do in the environment, we should be seeking other opportunities than geoengineering at this point. But I think people are finally catching on that there is climate change.
Mm-hmm.
Maybe in certain Republican circles, they're still haven't noticed the weather outside, but eventually even they'll catch on.
Mm-hmm.
It's a question, can we do stuff in time? CO2 levels, as you know, are getting to a point where we may hit the irreversible point.
Right.
So we're gambling with our lives. These microbes in the ocean provide the oxygen we breathe.
Yep.
We somehow kill those off. We're going to suffocate.
Yeah.
So those don't seem like smart choices to me. I don't know why. There's just something there that
I get it.
Seems like we can come up with better solutions.
Mhm.
But it's just a scale thing. It's
Sure.
and I understand it. People changing motor oil on their cars back in the 60s when we all used to do that ourselves. You think dumping that in the trash, dumping that in a stream, in the river, the ocean, you're one person, that's such a small amount, it's not going to impact anything.
Mhm.
Or you throw some trash out there. You multiply that by 6 billion,
Right.
and it's a hell of a lot of trash. It's a hell of
Yeah.
a lot of pollution. But we never think beyond our own individual spheres. If I throw this out there, it's just me throwing a bottle in the trash. But we have to think globally, because with the population so high, you can't do anything without people affecting the environment. When we're sailing across the Indian Ocean after the tsunami, we saw tens of thousands of flip-flops floating in the ocean.
Hmm.
Quite often these became rafts for sea snails and algae and all kinds of things. So they created new little sailing environments. But eventually the plastic and the rubber would break down and pollute the oceans. But just all the damage from the tsunami washed tens of thousands of flip-flops out into the sea.
What's I mean, I know you've been asked about I think looking in the other direction now. I mean, you've been asked about topics like terraforming in the past, and I've heard you talk about this. The European Space Agency's JUICE, or Jupiter Icy Moons Explorer mission, is headed out soon. It's supposed to get to Jupiter around 2031, and it's going to be checking out the ocean-bearing moons of Ganymede, Callisto, and Europa. Um, I don't know how Dinococcus will or tardigrades will do in some of those environments. And I know we have, per our recent episode with Ian Watson at the Pentagon, we got the planetary protection rules that, we got to be careful. No Andromeda strain stuff comes back down from missions, but that we don't contaminate unnecessarily parts of our solar system. That being said, is there a point where, maybe we put aside one of these moons to do some terraforming experiments? I mean, what are your thoughts on this? Because I know you talked in the past. I heard you think, concepts of like, sending one of your DNA devices out there to make some synthetic organisms on a planet. What are some of your
I think terraforming is potentially a nice romantic idea.
Mhm.
Unfortunately, we're doing it inadvertently and not wisely. So every time we send a probe to Mars, they're not sterile.
Yeah.
As much as NASA and SpaceX and other try to sterilize things, they're not sterile. So every time a new astronaut goes up to the space station, they're carrying with them millions of new microbes that haven't been seen up there before, especially with all the different countries and environments involved. We've sequenced the HEPA filters off the space station. They're not pretty. I mean, basically, the space station's a floating microbiology lab. But, we've already, I'm sure, introduced microbes onto Mars. It's a question of whether they can take hold or not. Once we get down to the subsurface oceans that supposedly exist, I expect that we'll find life in those oceans. The good thing about Mars is historically, as I understand it, we exchange about 100 kilograms of material a year between Earth and Mars.
Hmm.
And so, in theory, you can't take a shovelful of dirt on this planet without having Martian soil in it. and vice versa. So if we find organisms on Mars, they're going to resemble, if not be identical, to many of the ones we discovered on the Sorcerer expedition or other places, because of all those exchanges. You have to go out farther to Europa and other places, probably, but as we see, having life chemicals on virtually every meteor, I think life is going to be ubiquitous in the universe. But as we see with different environments, if we get a harmful virus in our bodies, that's not a good thing. So I'm glad there's planetary protection. I wish it was a little bit more active. Now that we have cowboys going into space, probably most of the rules aren't going to be followed too closely. The problem is it's difficult to get samples back from Mars. That's why I designed the biological teleporter, as you said, to put a sequencer on Mars, sequence the organisms up there, and send the DNA back as digital information and reconstitute the organisms here, like we did with the first synthetic organisms. But terraforming, introducing photosynthetic microbes to an ocean planet, if it doesn't already have them, that's how we got our oxygen atmosphere. Once we had an oxygen atmosphere, evolution took off. The thing is, those things don't happen in normal human lifespan. They happened over billions of years.
Right.
So it's not going to be a Star Trek version where you do a little explosion and all of a sudden you have palm trees. Right. But if we keep destroying the Earth, we're going to need someplace to move to.
Yeah. And while we're on this topic, you mentioned origin of life research, and there was it's near the end of the book. If I just can read this, you say, we're still trying to understand the interrelationship of the 20,000 genes in a human and the evolutionary steps it took to get there, or the steps that led to a sea lion or my dog to being alive and active and responsive to the environment. He said, right now, that's far beyond our current state of biology and meaningful intellectual pursuit. Say a few words, if you would. I watched your talk, your TED talk, in terms of speeding up evolution. Clearly, the ocean is full of creatures, whether it's the octopus or jellyfish or pond scum or whatever, that evolved in very different ways, whether, not just obviously morphology, but their brains or the way they process information. Obviously, life life is fascinating, goes in all different directions, but we really are at the beginning to understanding ultimately, what it takes to happen. Say a few words just about your thoughts on origin of life research and where a lot of what we're finding out in the work you've been doing the last 15 years per this mission is going to take us.
Well, in 2010, we made what we call the first synthetic cell. We chemically made the chromosome, starting with 4 bottles of chemicals, and then booted that up, and that converted the cell we put it into a totally new species. Mm-hmm. So then we went on to make, we designed a minimal cell, the smallest cell with the smallest number of genes in the genome. And it's been a great research tool. One of the problems with that minimal cell is roughly 20% of the genes were of unknown function. All we knew is if we took that gene out, the cell died. So our fundamental basic knowledge, we're still missing on the order of a quarter of essential biology. So we couldn't just do it by de novo design. We had to do trial and error, moving genes in and out. But eventually, as we understand more, we can do things by design. What the minimal cell is being used by scientists around the world is a starting point in evolution because I argue that we should be able to recapitulate all of cellular evolution by adding genes back to this minimal genome.
Mm-hmm.
And that's what scientists have done. Our own team with John Glass, the really basic minimal cell sort of resembles the soap bubbles that Jack Sozak's doing at the origin of life.
Yep.
And they were like blebs that divided. And we ended up adding 5 genes to the cell. And all of a sudden, the cell does normal cell division like all other cells on the planet do. Another team added genes for cellular mobility, and the cells started walking around the plate. So we should be able, as I said, to recapitulate evolution. And in each of these stages, for example, cell division, there were 2 cell division genes known. We put those in, they didn't do anything. We had to add 3 other unknown function genes to actually get it to work. So we're discovering functions as we try to recapitulate evolution. But if we can add things back, all of a sudden the cells are walking around, they can divide normally. We should be able to put pathways in. We should be able to make them photosynthetic, eventually even turn into a prokaryote, photosynthetic prokaryote, or even into a eukaryote, or maybe something like Methanococcus that makes methane. And once we do that, we're understanding the basis of life and the basis of actually doing life by design. But right now, it's an iterative process. Fortunately, there's major breakthroughs coming in the speed of writing the genetic code, because that was our rate limiting step.
Yep.
That's why it took over a decade to do of making these molecules by hand. So being able to write the genetic code will allow experiments to happen 10 times faster. And we should start to be able to truly replicate key parts of evolution to understand these basic cellular functions.
It's going to be fascinating next few decades for you. I got to tell you that.
It's like my goal with human longevity is be around to see it all.
Most definitely. Lastly, Craig, while we have you, anything that we should know about coming up as we just entering 2024 now? Conferences, other talks. I mean, obviously you're going to be back at the Institute studying these billions of organisms, but anything else on the horizon that I missed that is coming? I mean, I know the book's going to be launched in the EU coming up. Any other milestones that we should know about while we have you, please?
So the key other paradigm that we're working on is the reason I started human longevity. We're having trouble interpreting the human genome because the government funds lots of genome sequencing. It doesn't fund understanding of the genome by doing complete phenotype analysis on the people with those genomes.
Right.
So without having your genome, without knowing your physical characteristics, it makes it hard to learn anything new about your genome.
Sure.
So that's why I started human longevity, to do detailed, non-invasive phenotyping to compare back to the genome to move genome knowledge forward at a much faster pace. And we've had about 7,000 people go through, and one of the concepts is we only wanted to see people that were self-described as healthy.
Okay.
They couldn't have a pre-existing disease that they were aware of. And so I call this pre-symptomatic testing.
Mm-hmm.
And it turns out we find something in roughly 50 to 52% of everybody that thinks they're healthy. In 5% of everybody over 50, we find a major tumor that they were unaware that they had. This saved my life. It saved Ham Smith's life. Just having myself and my institute scientists go through we discovered each of us had a major tumor that had to be removed for us to continue living. So I'm alive today, and he's still alive in his 90s because of those discoveries. 1% of the population has a brain aneurysm, which, when you think of the size population, that's a lot of people. It's skewed to people under 60, which means brain aneurysms have played a selective role in killing off people that have them. Almost everybody knows somebody that's died from a brain aneurysm. They're treatable in about a 15-minute outpatient procedure now. So if you know you have it, it can be treated. Otherwise, you're waiting for it to pop, like a blowout on a tire. And usually people's first symptoms are a massive brain bleed that they go to the emergency room for and usually don't come back out. So we're finding a lot of pre-diabetes, a lot of metabolic syndrome. The MRI measures organ fat. That's the only thing that measures it accurately. So organ fat is one of the key indicators of health and longevity. I was on a 30-mile bike ride with the world's expert on organ fat. And we stopped for a break after 15 miles, and he goes, this isn't going to help reduce your organ fat at all. You have to restrict calories. That's the only thing that will reduce it. And I said, well, why didn't you tell me that 15 miles ago? So what we find is saving hundreds of lives and giving people the means to do more. We're now trying to extend this from people who can afford it to indigenous populations and underserved communities that can't afford it. So we've asked the government for help in funding it, some private donors, and we're trying to set up what we set up at Human Longevity with mobile MRI units and mobile clinics to go out to people who can't afford it. But it's a healthcare paradigm shift because we're looking for pre-existing conditions. You could have a tumor for 30 years before it manifests itself, right? But until you get symptoms, you're unaware that you have it. The medical community is unaware that you have it. But now the quality of MRI with the post-processing signaling makes it possible to detect a lot of tumors. It doesn't work yet for breast. It doesn't work yet for colon cancer, but it works for prostate, for brain tumors, for multiple other tumors that we find. So it's an exciting new paradigm, and it's helping to interpret the human genome. And we're trying to take it, it's the observation that Darwin made. By looking at isolated populations, you see isolated genetics, even though he didn't know what the genetics were back then. So it helps discover new bases of disease. Existing databases are largely Western European, Caucasian, and more Chinese, but they don't represent the populations of the US. And they certainly don't represent the indigenous populations that were originally the US. So we're trying to extend what we do in genomic medicine to these new areas. So that's a thing to keep watching out for.
Excellent. Yeah, no, it's refreshing to see the whole preventative and predictive structure with HLI, because we have been profiling a lot of the sort of the geroscience stuff, and a lot of it's a little sort of old school single magic bullet. But what you're describing here is extremely refreshing and needed. So no, it's great to hear that.
And it seems to be working. That's the key thing.
Yeah, absolutely. Really great stuff. I applaud you. It's been an honor listening to your stories and your journey, Craig. And I'm glad we had the opportunity to finally do this. And just really wishing you and the team there at the Institute the best with all this as you continue to move it forward. Again, for everybody who are going to be listening to this particular episode of our show across the various podcast networks or watching on the YouTube channel, again, you've been listening to Dr. Craig Venter, founder, chairman, CEO, J. Craig Vegner Institute. Pick up a copy of The Voyage of Saucer 2, available all major booksellers. It's an awesome read. Craig, again, thank you for taking the time out of your schedule. I know you just got over a cold, but really thank you for the time. Thank you for everything you've been doing. And as we like to say on our show, thanks for helping to create a better tomorrow for so many people via what you've been up to. It's a great story.
Thank you for your
sharing.
compliments. It's been great to spend time with you, and let's not wait quite so long to do it again.
Absolutely. Be well.